Yes, yes. So, ladies and gentlemen, good morning. It is my great pleasure to welcome you all to our online lecture series dedicated to exploring the wide diversity of life sciences. We are thrilled to have you join us for what promises to be an enlightening and inspiring journey through various scientific disciplines. So, in a world that is increasingly interconnected and rapidly evolving, the importance of science cannot be overstated. From the mysteries of the cosmos to the intricacies of the human brain, from the complexities of climate sciences to the wonders of technology, science touches every corner of existence. So, this lecture series has been thoughtfully designed to showcase the breadth and depth of scientific inquiry. So, now I would like to invite Dr. M. Prasanna Rajesh Kumar, Sir, Vice Principal, K.S.Rangasamy College of Arts and Science, to deliver the welcome address. So over to you, sir.
Thank you, Sati Babu. Good morning, everyone. With the blessings of line Dr. K.S.Rangasamy, sir, we have inaugurated this event. Beloved Chairman, Sir R. Srinivasan, sir, KSR Education Institution, respected Vice Chairman, Sir R. Sachin Srinivasan, our beloved Principal, Dr. V. Padmanabhan, distinguished speaker, deans, directors, heads, faculty, students, and participant of our KSRCAS and other college. Once again, a warm good morning, everyone gathered here. It is with a great pleasure that I welcome all of you to this exciting guest lecture series. Today, we have gathered here to engage with innovative ideas, gain insight from a distinguished speaker, and expand our perspectives on topics that are both timely and relevant. We are honored to have with us Dr. Umesh, Professor and Head, Research Cell Institute, Research Cell Institute of Management Studies, Ghaziabad, Uttar Pradesh, a renowned expert in oncology, whose work has inspired countless individuals and contributed to the field. He will be sharing he will be sharing his knowledge and experience with us, and we are truly privileged to have him here. This lecture series is an opportunity for all of us to enrich our understandings and spark meaningful discussions. We encourage you to take full advantage of this experience, ask questions, and connect with our resource person and each other to foster a collaborative learning environment. On behalf of KSRCAS, I would like to thank Dr. Umesh, Professor Head Research Cell, Ghaziabad, Uttar Pradesh, for taking the time to be here and sharing his valuable expertise with us. We are confident that this session will be both meaningful, enlightening, and thought-provoking session. Once again, I welcome and thank you all being part of this event. We look forward to a stimulating and inspiring session. Thank you.
Thank you.
Thank you, sir. Thank you so much, sir. Now I would like to welcome our beloved principal, Dr. V. Padmanabhan, K.S.Rangasamy College of Arts and Science, and patron of this event to deliver this presidential address. So welcome you, sir.
Thank you. Most respected guest of the day. Am I audible, sir?
Yes, sir. You are audible, sir.
Most respected guest of the day, Dr. Professor Umesh Kumar, Professor and Head, Research Cell, IMS from Uttar Pradesh. I think on behalf of K.S.Rangasamy College of Arts and Science, on behalf of our respectful management and the faculty and students team of K.S.Rangasamy College of Arts and Science, once again, as a principal of this institution, I extend my warm welcome to you, sir. Thank you for accepting our invitation and being with us today to enlighten our teachers and our beloved students on the area, especially like the emerging techniques in viral oncology related topics. So it's really, I feel really privileged to take part in this online lecture series. Yes, sir. it is the need of the hour that we need to share our knowledge with almost with our students as well as to our faculty community to strengthen the academic environment, because sharing of knowledge and sharing of transforming of our informations or our subject content may support in all means, sir. So I think at this juncture, I extend my congratulations and best wishes to the organizers, especially Professor Dr. G. Saravanan, Director, Research and Development and Head Department of Biochemistry, and Dr. V. Satya Babu, Assistant Professor of Biochemistry, and team Biochemistry, faculty and students for taking this wonderful initiation and bringing the experts here and making this as an interactive session with our students. It's really it's a happy moment on this particular occasion. Over to thank you very much, sir, and thank you very much for
Thank you
accepting
very
our invitation once
Thank
again.
you so much. Thank you so much. My
Yes, sir.
regards.
Yes, sir. Yes, sir. Yes, sir.
Thank,
Yes, sir.
thank
Thank you
you,
all.
sir.
Have a great day. Thank you. Thank you.
Thank you, sir. Thank you for your kind words. Now it's time to introduce the resource person, followed by the lectured by the resource person. So, ladies and gentlemen, it is my great pleasure to introduce our esteemed speaker today, Dr. Umesh Kumar, currently working as a professor and head in Research Cell, School of Biosciences, Institute of Management Studies, Ghaziabad, University Courses Campus, Uttar Pradesh. So he has done his PhD entitled Epigenetic Regulations in Breast Carcinogenesis from University of Delhi. Dr. Umesh Kumar has worked in Institute of Cytology and Preventive Oncology, ICMR, Noida. After his PhD, he joined in stem cells biology laboratory for his postdoctoral studies in National Institute of Immunology, New Delhi in 2014, where his scientific group has published research in the field of epithelial ovarian cancer stem cell in Nature Oncology, which is one of the prestigious journal in the Nature groups. And later, he worked in Department of Biochemistry, University of Delhi as scientific officer. Then he joined in Drug Discovery and Development Laboratory, Delhi University. So Dr. Umesh Kumar is a distinguished researcher with an exceptional track record of achievements and contributions across multiple scientific disciplines. He has honored with several prestigious awards, including the Outstanding Researcher Award, Senior Scientist Award, Scientist of the Year, Young Scientist Award, National Green Award, and the ICMR Senior Research Fellowship. These accolades reflect his profound impact on the field and his commitment to advancing scientific knowledge. Dr. Umesh Kumar has an extensive publication record of more than 50 publications in renowned international peer-reviewed journals with high impact factors. His scholarly work includes numerous research papers as well as contributions to books and book chapters. In addition, he holds multiple patents underscoring his innovative approach and contributions to science and technology. A recognized authority in his field, Dr. Umesh Kumar is also a distinguished speaker. He has delivered a multitude of lectures as a resource person at various esteemed institutions and conferences across India, sharing his expertise and insights with the broader scientific community. Dr. Umesh Kumar's research spans several key areas, including cancer biology, molecular epigenetics, molecular biology, molecular medicine, stem cell biology, translational oncology, molecular diagnostics, and recombinant DNA technology. His work in these fields is characterized by a deep commitment to advancing knowledge and improving therapeutic strategies, solidifying his reputation as a leader in contemporary scientific research. So we are truly honored to have Dr. Umesh Kumar with us today. His extensive expertise and remarkable contribution to the field make him an invaluable addition to our session. So I am confident that Dr. Umesh Kumar's presentation will not only be enlightening but also inspiring for us all. Without further ado, I now hand over the session to Dr. Umesh Kumar. Sir, the session is yours now, sir.
Thank you very much. Thank you very much, Dr. Shathi.
Yes.
I'm really thankful to the organizers who have given me this opportunity to speak up again in front of your faculties, scholars, and students. I would like to greet Principal Sir of this KSR Institute and Dr. Prasanna as well. Dr. Sarvanan is, a closest friend, and Dr. Sathi Babu, who is, working hard for the community. And see, this is a great initiative by Dr. Sathi Babu, where he is putting every efforts to accumulate all the knowledge in the form of different resource persons and different subjective experts. I would like to congratulate him on this occasion. And
Thank you, sir. Thank you so much.
yeah, and another that I would like to share among you, that you are really working hard to get those kind of exposure which we are getting in the laboratories to our students and scholars. So this is great, where a platform is given to your students and scholars. So I would also today discuss for these students and scholars those basic aspects. It is somewhat we as a scientist used to discuss different advanced molecular diagnostics and all these things, but being a moreover these students or scholars audience, I would be very pleased to introduce at basic level, and then we will go ahead with the topics. So I would like to share my slides. Just a second. I'm just, I'm taking that action where I can share my slides. Is it visible? Dr.
Yes,
Sathi,
sir.
is it
It is visible. Yes, sir. It is visible, sir.
it is full screen now?
Yes, sir.
And let me know if it is moving. It is moving.
No, sir. It's not.
It's not moving. Yeah.
Yes.
Just a second. I need to again reshare it, because let me share the whole screen actually. Entire screen I'm sharing. So is it visible now?
Yes, sir. Yes, sir.
The slides are
Yeah.
moving?
Yes, sir. Moving, sir.
Okay, okay.
Yes.
So today I have kept a very interesting topic, which will actually admire your students also. And if you go ahead with the topic viral oncology, like so that kind of the studies of that cancer which is happening due to the viral infections, and nowadays just because of the climatic changes and different level change challenges, lot of environmental issues are there, lot of pandemics, we came actually through a zone of the pandemic. So I'm just keeping this subject for the your scholars and students, because this is our challenge. uh, I mean someone's mic on. can
Ma'am,
you
ಹಂಗೆ ಬರ್ತೀರಾ?
can you off this mic?
Dear participant, kindly unmute your mics. Dear participants,
Kindly mute your mics.
so please proceed, sir. Hello, sir. Hello, can you hear me, sir? அவர் அன்மியூட்ல இருக்கிறார். நம்ம மியூட் இது பண்ண மியூட் பண்ணியிருக்காரு. எஸ். ஹலோ சார்.
Am I audible, Sathi?
Yes, sir. Now audible, sir.
Now audible, huh?
Yes, sir.
Might be some issue. I'm again sharing my screen, yeah?
Yes, sir.
Okay.
So, dear participants, so kind request, kindly unmute your mute your mics. So, if you have any questions at the end of the session, you can ask. Please note down the questions, and at the end of the session, you can ask. Now, kindly mute your mics.
Now, is it there? Is it visible?
Yes, sir. It is visible.
Yeah. Please let me know if any connection issue is there, and so that I can get aware. So
Yes,
I
sir.
was
Okay, sir.
discussing about those factors, how this cancer is occurring. So this is a multi-step process, and there is involvement of several genetic defects. There are several environmental factors, several risk factors, so diet also. So it generally taking some years to develop, so from this early stage to the final stages. So we should get actually the actual picture how it is happening. So it takes time. So that is a golden period to diagnose, at what level it is being diagnosed. Like, if this cancer is occurring due to some mutations or some epigenetic alterations, or there might be association of several microbes, right? It is very important to know. So with the development of these modern biological technologies, so it is now possible to diagnose exactly, and the exact subtype of the tumors and why it is happening. So this cancer can occur in any part of the body. So forget those things, like why it is happening. It can occur to any part of the body. Sati, can you mute the participants? You must be having some control. There, yeah. Thank you. So this cancer is generally, particularly an old age disease, but nowadays, due to several risk factors, it is occurring, too early in the different age groups. So, and the good thing is that majority of cancers can be prevented, or that can be curable if detected at early stages. So it is important to know about those risk factors, which is actually allowing to develop a, effective preventive measures. So strategy is to diagnose at early level. So that kind of awareness is very important. If we are not able to diagnose timely, so that eventually develop at the later stages, and which becomes very difficult to diagnose it, right? So we should know about the biology of these pre-malignant lesions, which is happening, which has been happening in the due course, and we should know that key to developing those effective preventive methods, just because of this understanding of the biology. So which individuals are at risk, this is important, and for what type of cancer, this is important. What are the best methods of identifying and monitoring these pre-malignant diseases, and how can we safely and effectively arrest the progression of the pre-malignant disease. So we should prevent to go to progress that disease from early stages to malignant diseases. So this is very important, and we should focus on what are those causes of this cancer. So all these cancers can occur through carcinogens, and those carcinogens could be any chemicals, could be viruses, or could be radiation. So we already know that radiation used to suppress the apoptosis, and we already know there are several carcinogens which are actually causing the disease. And viruses, there are 10 to 15% cancers which are occurring through viruses, and these viruses I will go into the detail in coming slides how it is happening. There another challenge is, I mean, the causes of the cancer is hereditary, which is also 10 to 15% cancers are associated, like breast cancer and some other lung cancer also. So there are genetic defects which can be detect in the patient, and then we can follow up with the whole family with those kind of suppressor genes, susceptible genes, and in combinations also it can occur. Sporadic cancers can also occur, right? So almost all human diseases result from interaction of genetic susceptibility factors and environmental factors. Now this is very important to all the genetic susceptibility is associated with the environmental factors also, right? So e.g. infections of viruses, bacteria, chemicals, physical, nutritional, and behavioral factors. So anything and everything outside the body that interact with human as environment, and this includes lifestyle factors, diet, tobacco, alcohol, radiation, infectious agents, and substances present in the air, water, and soil. So now we know that this cancer can occur, in different types or different methods or different ways, right? It is associated with the environmental factors also, and it is also having some susceptibility genes inside the body. So there is a mechanism to develop a cancers. So it is not a single process. So I would not go much onto this slide. Components of environment. There are everybody knows that air, water, soil, housing, climate, geography, heat, height, noise, debris, radiation, all is physical. And then we have biological component, human, viruses, microbial agents, insects, rodents, animals, and plants. Everything is dependent in each other. Psychosocial, that is cultural values, and there are several other: education, religion, morals, attitudes. All these are components of environment. But this radiation and uh, chemical pathogens and pesticide, smokes and pollution, this is affecting all overall health of these animals and plant and environmental health, which is actually suppressing those tumor suppressor genes and developing several diseases, not only cancer, several diseases, and ultimately affecting the human health and making the human health burden in India, right? So I told you that almost 80 to 90% of human cancers is because of these environmental factors. So my primary aim is to identify those specific microbes, either in form of viruses or in the form of bacteria, which is actually causing cancers, right? So cancer is a multi-step process. I already told you that it takes several times to develop this cancer. It start with some inflammation, and then it actually used to promote, and it used to progress with the several mechanisms, and finally it is it comes in the forms of metastasis. So infectious agents, which we are discussing, and substances present in the air, water, and soil, that actually how it is actually affecting cancer. So an individual may have a genetic mutation, that could be other possibilities. Like suppose a person has a gene that increase the chance of developing melanoma. So melanoma gene and sunlight exposure, that is actually coming in the outcome of cancer. Why? Melanoma gene is already susceptible, right? So it is that the risk of melanoma is 50%, but when it comes in the combination of sunlight exposure, so it becomes more prevalent, that that the chances of cancer development is more. So this is how we have to see that how are we exposed to these environmental agents. That is why we need to know about the roots of those exposures, like air we breathe, it is coming through air; food we eat and beverages we drink; contact with our skin, and a contact with the eyes. Some placenta, some can appear in breast milk, exposure to infections, Kindly mute yourself, participants. I would be happy. Yeah. So these environmental factors and the involvement of the cancer deaths in the percentage form you can see. So if you go with the those exposures, so if you see the tobacco diet, tobacco is 35%, but infection is almost 10%. So this is our concern, that which can be prevented, right? So we cannot deny that the whole population is having exposure of a lot of people are having exposure of tobacco, and the kind of other risk factors, pollution, we cannot control that level. So it is now everywhere in metro cities also, and our sedentary lifestyle and obesity is also occurring, alcohol consumption is there. So all these depends, you know but our focus should be on those preventable things which we can prevent, right? So infection is also there. So viral and bacterial infections which actually leading to the tumors, we can control them, and we can develop some vaccines also, preventive level vaccines also. So there are several factors associated. One is internal factors like hormones, ER, PR, HER2/neu. These used to get suppressed in case of breast cancer and ovarian tumors. Immune conditions that these days autoimmune diseases are occurring, so it is more prone to have tumors. Inherited conditions like some genetic makeup is going on within the family, like BRCA1 and BRCA2 genes in the breast cancer. And then those are external factors like smoking, diet, and alcohol, viruses, chemicals, radiations, and other environmental factors. So these causes different types of cancers. So a major risk factor is viruses. So like human papillomavirus, hepatitis B and C virus, and in bacteria, Helicobacter pylori, which is actually causing intestine tumor, right? So uh, all these risk factors are actually associated with developing those cancers. So human papillomavirus, as you know, that it is affecting majorly majorly two types of this those high risk in India, HPV 16 and 18, which is causing cervical tumors in the woman, right? So it is number 2 cancer nowadays, just because of the awareness. Earlier it was number 1 tumor in early 2000s. So due to this awareness and development of several preventive and therapeutic vaccines, so it is now coming down, but still there is a lack of awareness among the population in particularly tribal regions. So it is occurring more in those regions, and there are other environmental and exposures like radiation, radon, asbestos, organic vapors, and pesticide is also nowadays very concerned in developing those tumors. So these risk factors and involved in different tumors. So it would be interesting to know that nearly all cancers of lung, bladder, mouth, and skin could be prevented, right? So pesticide is main in the lungs and these mouth cancer and skin cancer. 50 to 75% of cancer deaths are related to personal behaviors. I mean, those social activities and or habits. 30% or more of all cancer deaths related to cigarette smoking. 30% of all cancer deaths in the US are related to poor nutrition and inactivity leading to obesity. Risk may be reduced by increasing consumption of fruits and vegetables and limiting high-fat foods. So these are those factors which should be very in the mind that we should reduce those risks actually. Our motto should be those things. So here are those important biological agents which I'm going to discuss. Associated with these different tumors. So these are those, like human papillomavirus, I told you, cervical cancer, in anogenital cancer, skin cancer, oral cancer, esophageal cancer, oropharynx cancers. So in our previous study where we were studying the Northeast belt, so the percentage of human papillomavirus positivity is high in the anogenital and oral cancer in northeast, while in Kashmir, Jammu Kashmir, esophageal cancers are more, and the positivity of HPV is also high. Um, Epstein-Barr virus is very important, which is causing non-Hodgkin's lymphoma, nasopharyngeal carcinoma, Burkitt's lymphoma, Hodgkin's lymphoma, Kaposi sarcoma, primary effusion lymphoma, HTLV1. This is also causing some T-cell lymphoma, adult T-cell leukemia. So HIV, HHV, in combination, they are creating in different tumors. They are involved, right? Multiple myeloma, lung conjunctivital, squamous cell carcinoma, cervical carcinoma, testicular carcinoma, hepatitis B. Everybody knows that it is directly related to liver tumors, right? Liver cirrhosis. So there are involvement of several viruses, and Helicobacter pylori is very important. I mean, there was a recent Nobel laureate award from Australia in the while for this Helicobacter pylori. So this causes stomach cancer, lymphoma, cystostoma, hematobium, bladder carcinoma. So there are involvement of several biological agents, and this could be prevented. This could be targeted, so by using some several, either vaccines or either some therapeutics. So and if it is arising a lot these days, because this is not a ratio of India, in Southeast Asia, a lot of infections are occurring, which is causing actually tumors, such type of infections. So these risk factors which we are discussing, those genes which known to be involved or suspected of being involved in these familial cancers like BRCA1 and BRCA2, in case of tumor, the breast cancers, and interactions of the genes with the lifestyle factors, environmental and occupational exposure. So this in combination, it is causing a complex tumors. So there is a aflatoxin, which is a fungi which is growing into the stored food grains, that is also causing some liver tumors. So we should be aware about these risk factors. And then there's some physical inactivity nowadays, it is happening, unsafe sex, diet, there are a lot of carbs involved these days, alcohol use, it is very social factor nowadays. And you see that the pesticide is also another factor which is causing that cancer risk. A higher cancer risk in the male farmers, like lip cancer, skin cancer, stomach, brain, lymphoma, prostate, particularly in Northeast Indian, because we have studies that belt of Silchar, Dibrugarh, Guwahati. We took samples from there of different origins. So we have studied the role of tobacco and pesticide in specific regions in ICMR study. So now this is very important, to know, because this is my area, how this infection of human papillomavirus is causing cervical cancer, and how we can prevent. There are some easy methods where to go once it is being developed. What is the severity of the disease, right? There are several risk factors. This is for the awareness of those girls which are listening, right? So these are those risk factors which are associated to the cervical cancer, which can involve human papillomavirus infections. These human papillomavirus infections are, this is a almost 8 kb virus, which is having two consensus regions, right? This gives an idea to diagnose this on the basis of these the L1 and L2 consensus regions. But here we are discussing those common risk factors which you should get aware, right? There is a risk factor with the early age of first sexual intercourse. If it is less than 18 years, so the earlier sexual activities also giving chance to develop this HPV infection. And then there are multiple sexual partners, sexual activities, poor genital and sexual hygiene. This is also the in the early ages, the women were not using those pads. They were using the cloths. That poor hygiene was the main cause of this HPV infection. Number of pregnancies are higher, oral contraceptive use, and then smoking, and these genital warts. This can be reported actually. Warts can be anywhere, but these genital warts can be severe. It comes with the HPV infection. Then ultimately There are involvement of the dietary factors also, low nutrition diet, and these are other factors which comes in the studies, right? The education, socio-economic status, ethnicity, where it belongs, and infections of other STDs can also leads to several other opportunistic infections. So cervical cancer is preventable cancer if detected early. It is very important because once it is detected, the overall survival rate goes to very low. So the risk factors are controllable, as all these factors are intimately linked with lifestyle and behavior, right? So this could be prevented. We already know that there are several vaccines available nowadays, and that people are getting aware about this. Adults and girls are taking these kind of vaccines which are available in the market. Gardasil from Merck and the Cervarix from GSK. So these are four different subtypes. I'll discuss these subtypes because I worked a lot on HPV 16 and 18. So there are major high risk HPV 16 and 18, 6 and 11. So this is a quadrivalent Gardasil. That means that this will tackle with these type of subtypes, HPV subtypes 16, 18 and 6 and 11, right? So we can choose according to those kind of action against those kind of viruses. To And this is a time specific. This is given from 0 to 13 years. So nowadays it is easily can be discussed with the gynae before taking this. This is very safe, 99.99%. Giving the specificity, right? So what can we do to prevent these cancers? We can be good with the healthy diet, exercise regularly, maintain healthy weight. We should not smoke, alcohol in moderation, increase fiber and reduce fat. So know your risk and seek appropriate evaluation or therapy for your risk levels. What should be our goal? Reduce fear and misinformation. In the time of the science, we should refrain to those misinformation or WhatsApp information through cancer information and education, proper education, increase awareness of cancer prevention, increase screening and early detection strategies, increase lifestyle choices, and that may be reduce cancer risk, and describe source of information and support if you have questions, right? So early detection should be our aim. Screening for cancer means looking for cancer before there are symptoms, right? We should be alert for that thing, which whatever risk is involved. We should actually have that kind of knowledge that something is being developing and we have seen some nodes or some warts. So we should be careful. Once it is there, we should go immediately to the clinicians. Screening may identify early case of cancer that might never have become clinically apparent, and that kind of cancer prevention involved primary prevention, secondary prevention. At least 50% of cancer can be prevented. I already told that primary prevention efforts have the greatest potential to reduce the burden of cancer, right? So remove the cause of cancer, whatever the risk factor. Screening is very important. At and these days, at sometime at primary level it is difficult to diagnose, but they used to send to the secondary or tertiary center once it is being suspected. Medical or nutritional treatment that prevents the development of cancer. A lot of yoga practices and Ayurveda and lot of alternative therapies can also assist in the treatment. While discussing with the primary prevention, so we can remove those causes, reducing, avoiding personal exposure to those carcinogens in the environment, reducing or eliminating the release of carcinogens into the environment. And then we have personal health behaviors. We should refrain from tobacco, assign exposures. Obese peoples are more prone to have tumors, right? So healthy lifestyle. And these are those modifiable risk factors like primary prevention, healthy diet, exercise, uh, and secondary prevention, like we used to train those women also in our clinical practices also. Breast self examinations, where we can find out some node, some node positivity, or kind of some tumors, right? Testicular self examination. There is a mammography which can be preferred to for some parts of the body. Skin examinations also. Pap smears we can collect for cervical cancer positivity, and there are PSA test also. Colonoscopy can be done for anogenital anal cancers. Fine needle aspiration cytology, which is being done in several tumors like breast examinations. And there are some other screening tests like thyroid and liver function tests, cardiac function tests with this, right? So diet and nutrients actually protecting from cancer. So in the current era, it is very important. So now we should, improve our diet. We should include those fruits and vegetables, which is high in fibers, which is carrying antioxidants. I know that this is also area of Dr. Sathi Babu. All these natural phytochemicals, which is actually playing a role in the preventing the different types of tumors also, or in the management of the disease also. Tocopherol, beta carotene, lycopene, curcumin, green tea, red grapes, garlic and onions. They can actually, play at molecular levels. They can actually activate those markers like glutathione transferase, which is very important isoenzyme, which is actually helping in the conjugation of carcinogens with cellular glutathiones. So all these diet and fruit and vegetables can actually be helpful in protecting from this cancer. There are cruciferous vegetables, omega-3 fatty acids, fish oil, soy products also, right? So these isoflavonoids can compete with estrogen for its peripheral receptors on breast, blocking them. So every individual thing which is actually playing at molecular level. So we should actually be very aware about these red grapes, which is containing resveratrol, and turmeric. I already told curcumin. Curcumin is very good anti-tumor, but in moderate amount, right? So tumor cells secrete factors that promotes the development of new blood vessels, which are necessary for tumor growth. So this process is known as angiogenesis. Resveratrol and curcumin suppress the release of growth factors by tumor inhibit and angiogenesis, right? So all these, bioactive compounds can be helpful in the managing the disease also, or preventing from these diseases also, right? Prevention, we should focus on the early screening and prevention from the cancer. There is a no treatment which we can tell. This is only the management of the disease, right? We do not know how the individual body is behaving after giving those kind of treatment. So it is just the management of the disease, right? So this is my laboratory here in my campus, and my scholars are there, which are really working hard in CAR T cell therapy, stem cell therapies, right? So I would be, on the basics only. I could have gone how this molecular diagnosis can be done. I do have those slides. If times allow, I can speak. But I do have a meeting also. I have to go. But I'm giving those kind of basic things. How these cancers can be prevented, how these cancer can be screened, right? So Sathi Babu, are you there?
Hello.
Sathi, are you there?
Yes, sir. Yes, sir.
Yeah. If I can give some ideas of some screening methods, I want to share another a PPT for 5 minutes only, if you allow, right? Because
Yes,
these
sir.
were
You
the
can.
basic who the cancer
You
is
can,
occurring
sir.
and what are the risk factors. So
Yes, sir.
I'm
Please,
just
sir.
keeping those slides short because I do have a meeting. I'm quickly sharing that, right?
Yes, sir.
Just
Please, sir.
So I wanted to share this basic slide. I want to show you that how these diseases being developed, different stages being developed. That takes, I was telling that it takes years to develop a cancer, 10 to 15 years approx, from normal cervical epithelium to invasive cancer. So this is very important to screen actually. I want to tell you that human papillomavirus, it is a 8 kb virus, right? And there are low risk HPVs.
Dear participants, so there is a technical issue from the resource person. Please stay tuned. He'll be back.
Sorry, there was a disconnection. Dr. Sathi, am I audible?
Yes, sir, you are audible, sir. Now you can share your PPT, sir.
Sorry, actually, somehow I disconnected.
No issue.
On wrong button. So I'm
Yes.
sharing again those basic slides, how to diagnose a HPV virus, right? Let me know this slide is visible to you, the basic
Yes, sir.
genomic
Yes,
organization of
sir. Yes, sir.
HPV.
Yes, sir. It is visible, sir.
So quickly I'm going because I have a meeting also.
Yes, sir.
I'm using a pointer to tell the students or scholars that this virus is of 8 kb, which is having that two consensus region L1 and L2. So this is a basic target to diagnose this virus at genomic level. We used to amplify these consensus regions using PCR. So this gives an idea about that which type of virus, is there. That means HPV is positive or not using PCR. But once this is diagnosed, we have to go with the which type of specific HPV is there, either HPV 16, 18 or something like that. I already discussed about the risk factors. I'm going with those diagnostic methods, like we were discussing. There are methods, cytological methods. We can have a pap smear and we can diagnose using those cells which are in pap smears. There are pathological diagnosis like IHC, and then HPV DNA test, which we used to do. DNA based testing is premier testing in HPV to confirm it. There are involvement of primary screening methods. Once these are done, then we used to go with the HPV DNA diagnostics, right? So PEP test, everybody know Pepinekulo stained smear method. We used to have those swabs in the zones of those gate of the cervix, and we used to exfoliate those cells into the PBS, and then we used to take those pap smears to the labs, and this gives an idea about the classification of those tumors. Either it is squamous, high grade, HCl, as HCl, LCl. So this can give the idea, which stage is there, early stage is there or later stage is there, right? This is very important. Yeah. So there could be limitations of cytological screening. That is why we used to go for the further test, ASC test, USC. ASC means like, this is, done for these colposcopy tests, visual inspections also. If it is fails, then cervicography also. So everyone is having limitations, but once it is being suspected, then we should go for the molecular testings, and that is detecting viral genome. That means the DNA of that virus. So we should go with the rapid or simple, cost-effective, sensitive, and specific, reliable, and reproducible test, which is PCR, which is PCR-based ELISA or hybrid capture tool. These days this is very common. Hybrid capture tool is
Sir, you're muted, sir. Hello? Hello? Hello, Umesh, sir?
yeah. Is it audible?
Yes, sir. Now we're audible.
Now audible. So I'm
Yes,
again
sir. Yes.
sharing. I
Yes,
was
sir.
telling those screening methods, possible screening method. HPV is diagnosed at DNA level, right? So these are at least 10, methods of those detection. We used to do the PCR and HC2 and even sometimes real-time PCRs. So these are different techniques which can be used in the laboratory. This is for your scholars, which types of methods we can use, and the which types of results we can get, just using the southern blot, either in situ hybridization, fish dot blot hybridizations, and some cytology also, right? So in situ hybridizations. So we can go with different things, but here these are very important as far as HPV is concerned. So PCR L1 is specific test, E6 and E7 regions spec. MY9, MY11 are universal primers for the HPV diagnosis. We can use these things. Multiplex PCR we can do of different subtypes of HPV. Real-time PCR will give an exact idea, and mRNA-based PCR, right? So these are those things which I wanted to tell in this slide. And the positivity you can get, like L1 positivity is there, HPV16 is positive there. This is beta globin, and then this is for HPV16 positivity, right? So these are those different types hybrid capture, which can give an idea of different subtypes of HPV test, right? High risk HPVs, okay. And then these are basic things. What is hybrid capture, right? That your students can go through with their thing. I already told that this is very important to collect and carefully storage those specimens to get positivity, right? Pap smear is the simplest method, primary method, and then we can go with the several DNA based test, molecular test, right? So this is my experience, where I used to find out in the urine also sometimes, right? Uh, there are some positivity can be done, but so cells are very inadequate. So Sathi Babu, I wanted to keep it short just because I have a meeting, right? And I already discussed about the screen strategies and vaccination strategies. We have discussed about those two vaccines also, which is are available. That is very important. So my concluding remark will be that this viral oncology is very important in the current era. Lot of viruses are occurring and they are actually mutating themselves and developing different subtypes. So these high risk viruses are very important to diagnose at timely in the pre-malignant lesions or pre-early regions, so that it can be detected timely. And the lot of burden is there. I mean, in women HPV is very much involved, and it causes second number tumor, that is cervical tumor. So I wanted to have those queries quickly, because I do have a meeting. So we can take one or two questions if you allow. Am I audible? Yeah. Is there anyone who can drop a either in the form of messages or any questions? Thank you so much, everyone.
Sir, good morning, sir. Myself, Dr. Viral Oncology, Assistant Professor of Biochemistry, KSRCAS.
Okay.
Sir, I had a question, sir.
Okay.
Sir, what is the maximum period for getting vaccine for this HPV, sir?
Your voice is breaking. Can you put in the message? Can you type your question? I can see over here, Subhasri, because a lot of noise disturbance is there. Can you type this question in type box, message box? I'm on the my message box. Yeah. There might be some networking issue, I guess. I heard that you were asking about some HPV diagnosis, but I couldn't understand the whole question. Dr. Sathi, can you explore that question?
Yes.
What is
Yes.
that question?
Ma'am, can you repeat the question or you type in a chat box, ma'am?
Yeah.
Dr. Viral Oncology, ma'am.
Sati,
Your voice is
is
breaking.
it audible?
Your voice is breaking, ma'am.
I'm putting my email ID. I can send you these slides to this. You can use this email ID. I can give those slides also, and I can give any questions on this also, right? But meanwhile, I'm taking one or two questions if you can put in the box.
Sir,
Yeah, I can see some.
There are some questions in chat box, sir, please.
Yeah, actually, I wanted to know how pediatric cancer is different from adult cancer and why it is of more concern. Yeah. So although this comes, I used to work on those women tumors, but still I want to reply on that. See, it is all about those different subtypes of tumors, right? In pediatric cancers, like in blood tumors, we can see that there might be several association at metabolism level, and that the metabolism rate is very fast in kids, and sometimes those susceptible genes are coming through womb. I mean, there might be chances of development of some mutations in the early age group, and there are specific tumor suppressor genes or oncogenes which are getting involved at that level, right? So although the stemness markers are there, but still those pediatric cancers involved a lot of involvement of the oncogenes, right? While in particular age group or old age group, there are involvement of the tumor suppressor genes which are getting silenced of different mechanisms. So we have to understand, there are several types of genes, oncogenes and tumor suppressor genes. As far as we are growing, there are chances that there are chances of getting silent of these tumor suppressor genes due to some mechanism. These involved epigenetic alterations also, mutations also, and several genetic recombinations also. But at early age group, while it is growing, so there are few cancers which are occurring due to those oncogenes overexpressions. So there is a difference, but the cancer is there. So this is a cells disease. So we have to restrict all on to those genetic information of that particular cell, and every cell is showing different expressions, right? Every organ cells. So we have to be tumor specific. We cannot compare these tumors. See, if we discuss about the breast cancer and other cancers, there are involvement at NF-kappa B and AP1 family also, right? If CGen, CFRAS, FRO, CJUN, these transcription factors are associated in these are overexpressed in particular cancer, it doesn't mean that it will overexpress in the another cancer also. So we have to go at that molecular level to make difference at at this level. We can commonly comment that there are involvement of several genes, oncogenes, proto-oncogenes at pediatric cancer, most probably BCL, ABL. These are those genes which are actually involved in the pediatric tumors, right? Any other question?
Yes. So thank you, sir. Sir, I have a question or a request, sir.
Yeah.
Actually, our students are in a phase of submitting research proposals for their project work, sir, to the Tamil Nadu State Council for Science and Technology. And most of the proposals are dealing with cancers. As you are renowned expert in the cancer field, so
Right.
can you give some suggestions to our students?
Yeah. My suggestions would be to work on preventable measures, because we are discussing about the treatment options, right? We are not going into the depth of the diagnosis. If the disease diagnosis is being achieved, accurate diagnosis, or you can say the accurate subtyping of the tumors as far as at molecular level, so we will be able to easily deal with that particular tumor. We should focus on the exact diagnosis or subtyping of the tumor. We should refer to those biomarkers. We should be aware about those biomarkers which is causing that disease. See, treatment is just a you know kind of step away. Treatment is just a step away when we are at the stage of diagnosis, accurate diagnosis. See, the things like epigenetic biomarkers are coming these days, right? So when we say that cancer is not a single step process, so this is how we are dealing with that. This cancer cell is having so many pathways. We have to see every pathway, and this can give an idea. A lot of techniques are there, by even DNA microarray is there. We can put that DNA microarray, or even whole genome sequences. We can go with the exosomes also. A lot of involvement are there. So I would suggest to work them onto the biomarkers, and also on the preventive methods, like we must include some Ayurveda and some homeopathy medicines which can actually regulate those cells at cell cycle level or some other level where the actual machinery is getting affected, right?
Yes, sir. Thank you. So, dear
Yes.
participant, is
Sir,
there any other questions?
Dr.
Only
Prasanna.
one.
Yeah,
Yeah.
Yes, sir.
Sir,
Please,
it was
please.
really a nice presentation, sir.
Thank you,
Just I
sir.
want to clarify one thing. Sir,
Yeah.
sometimes few human papilloma virus
Yeah.
infected person, they eventually develop a cervical cancer, sir. What is the reason for this, sir? Why it happens, sir? Not all HPV, but
Yeah.
especially a few human papilloma virus.
Yeah. That is a very good question, Dr. Prasanna. Actually, we have to go with the integration of that virus into the host genome, right? These kind of viruses, I already discussed about the risk factors, how it is coming. I do not know which could be the pathway, but once it is exposure to the epithelial tissues, it will certainly integrate into those epithelial cells, and it will go at genetic level. I mean, that gene will be there into our DNA, and now it will use now our host mechanism to manipulate, or to go further. So this kind of infection, that this changes with the stages wise, and the early stages will involve this kind of infection, but it gives times us to diagnose at that level. If we can diagnose at that level, it can be best managed. The problem is that we sometimes information is there, that there might be symptoms like bleeding in the cervix or something like that. So this could be from other conditions also, but thing is that we actually avoid those conditions, those symptoms. We should go to the immediately to the gynecology, and there is a VIA test, huh? So VIA test can give an idea about those those proliferated cells, or those cells becomes white when we apply those acetic acid or sirca test. This is very common over here in our local regions. This is a primary method, VIA test. So this can give idea if it is cells are epithelial cells are becoming that inflamed cells. So inflammation actually leading to tumor. So this is the golden period where we can diagnose. As far as your concern is your question is concerned, HPV used to integrate into the epithelial cells, right? And it ultimately developed tumor in stages, not like the day one or day first year or second year. It approximately takes 7 to 8 years to develop that tumor. So this is a golden period for us. Once it is happening, now we do not know which type of therapies we should use, right? This is a very complex region. We cannot surgically eradicate those tissues, right? uh, Nearby organs are there, ovaries are there, uterus are there. So it will certainly affect at metastasis level also, right? It will lead to even liver or lungs also.
Okay, sir. Thank you. Thank you, sir. Thank you.
Thank you. Somehow my camera is not working at this stage. I was on that webcam somehow. Huh? So I would be happy to take more questions right now. Then I have to go into the meeting, right?
Yes.
Is there any questions in
Thank
chat?
you, sir. So participants, resource person has to go for some other engagement. So if you have any questions, you feel free to contact through mail. So he's always ready to answer you. And thank you, sir. It was a great session. It's a time to thank the people who are all involved in this event. So now I would like to invite Dr. S. Vadivakrasi, Assistant Professor, Department of Biochemistry, K.S.Rangasamy College of Arts and Science, to propose vote of thanks.
Myself, Dr. S. Vadivakrasi. Good morning to everyone. It is both an honor and a privilege for me today as we come to the close of what has been an inspiring and impactful session of Day 1, special online lecture series jointly organized by Departments of Biochemistry, Microbiology, Biotechnology and R&D Cell, K.S.Rangasamy College of Arts and Science Autonomous, Tiruchengode. On behalf of organizing committee, I would like to extend my heartfelt gratitude to all those who have made this a very possible one. First and foremost, our deepest thanks go to late Lion Dr. K.S.Rangasamy MJF, the esteemed founder, and to Mr. R. Srinivasan, the chairman of KSR Educational Institutions. We owe a debt of gratitude for your steadfast guidance and support. Our appreciation extends to Mr. Sachin Srinivasan, the Vice Chairman of KSR Educational Institutions, and Mrs. Kavita Srinivasan, Executive Director, KSRCAS. We are also deeply grateful to Dr. V. Padmanabhan, the Principal of KSRCAS and a Patron of KSRCAS. Your support and encouragement have been pivotal in fostering a nurturing and progressive environment. I want to extend my gratitude to Dr. M. Prasanna Rajesh Kumar, Vice Principal, and Dr. G. Saravanan, Dean, School of Sciences and Humanities at KSRCAS. On behalf of organizing committee, I would like to extend our heartfelt gratitude to Dr. Umesh Kumar for gracing us with their presence today. Your insightful presentation has truly been enlightening. Your depth of knowledge and passion for the subject matter was evident and has undoubtedly enriched our understanding. Thank you, sir. We further extend our sincere thanks to Dr. Suresh, librarian KSRCAS, for providing necessary online meeting Microsoft Team facility for this event. In closing, I want to extend my gratitude to all the attendees, participants, and contributors who have been part of today's success. Your presence and engagement are what makes these endeavors meaningful and impactful. Thank you once again to everyone who has played a role in making this session a memorable one. We look forward to day 2 with renewed enthusiasm and a shared commitment to excellence. Goodbye for today and meet you tomorrow. Thank you all.
Thank you, ma'am. Thank you so much. Dear participants, now it's time to signing off day 1, and we are looking forward to day 2. And kind information that day 2 will be start tomorrow 10:45 a.m. So kindly join with us and have a great day. Thank you so much. Now it's time to leave. So thank you so much, sir.